This is part 5 of the Chapter 5 lecture, and as I'm recording this, I have to apologize for how very long it is. But as you can see, there is a lot of detail in this chapter that it hopefully is review, so you can quickly run through this. But we left off speaking about proteins and the different roles that they play. So again, they account for more than 50% of the dry mass of most cells, and they are essential to life. Their jobs include structural support, storage, cellular transport, cellular communication, movement, and defense against foreign substances. And probably the most important function is their role as enzymes or catalysts in cells, because they regulate metabolism by selectively accelerating certain reactions, and they're not consumed in those reactions. As humans, we have tens of thousands of different proteins, and each of them has a specific structure and function. So that structure, again, it comes down to a monomer and a polymer. The monomer is an amino acid, and there are 20 different amino acids. And a polymer is when we have a series of amino acids joined together into a polypeptide. So a protein can be made up of just one polypeptide chain, or it can be several polypeptide chains bonded together. They can be very large and complex molecules with very complex three-dimensional shapes. Okay, so starting with the monomer, the amino acid structure you should be very familiar with. So there's a central carbon here, asymmetrical carbon, at the center of amino acid monomer. This carbon atom is called the alpha carbon, and there are 4 elements that are attached to this carbon. We have at 12 o'clock a hydrogen atom, and then at 3 o'clock we have a carboxyl group. At 6 o'clock we have an R group, and at 9 o'clock we have an amino group. So those R groups are actually what define the amino acids and make the 20 different amino acids what they are. And they each have chemical properties that determine the shape of the protein, actually. So we group them into different chemical properties, starting with these that are nonpolar or hydrophobic. So as you look at the white boxes are your R groups, and you need to just be familiar. You don't have to memorize the structure of each of the 20 amino acids, but you do need to be familiar with what would make it nonpolar and hydrophobic. And if you look at these, there are 2 possibilities. Either you have a carbon-hydrogen makeup, so hydrocarbon makeup of your R group, or you have a ring structure. Both of these are very stable structures, and so they end up making these nonpolar, non-reactive, and therefore they are hydrophobic. The next group are the both polar and the hydrophilic groups. So these ones, you need to look again at what makes them the way that they are. And you'll see that what is most likely present is either oxygen or nitrogen. So the oxygen present will make them, particularly in these groups, acidic, and the nitrogens will make them basic because the nitrogens can accept hydrogen protons, and the acidic groups can accept hydrogen protons, or actually the electrons. So again, just be familiar with what these different polar and electrically charged groups have in common. Okay, some acidic R groups have a negative charge due to the presence of a carboxyl group. So you can see the presence of the carboxyl group here. And these R groups have a positive charge because they are, again, proton acceptors. And you can see the presence of the aminos there, the amino groups. The last group are those that contain sulfur. So there are two R groups or two amino acids that have sulfur in their R group, and these two are the ones that can form disulfide bridges, which is a covalent cross-link between sulfidyls. And these are very stabilizing bonds. They're probably the strongest bonds that hold, aside from the polypeptide chain, to hold a protein structure together. Okay, so how do we go from the monomer of that amino acid into a polypeptide chain? We do that through dehydration synthesis again, forming peptide bonds. Those peptide bonds are the strongest bonds that are holding the protein together. They're covalent between the amino group of one amino acid and the carboxyl group of another, and they form this carbon-nitrogen covalent bond. And again, remember that dehydration means you're going to remove one molecule of water in order to form this bond. So this repeating process creates a polypeptide chain. When we're describing this polymerization of a protein, what we say is that at one end of the amino acid, there's a free amino group, and that's called the N-terminus. And at the other end of the amino acid is the free carboxyl group called the C-terminus. And that's what's forming this peptide bond. And polypeptides can range in size from a few monomers. You can have a dipeptide with just two amino acids to thousands of polypeptides long. But each polypeptide has a unique linear sequence of amino acids. And this is just reiterating the N-terminus and C-terminus, and the repeated sequence makes up the polypeptide backbone, or the primary structure of your polypeptide. So now moving on to what happens after we form that primary structure, that chain of amino acids. Frederick Sanger and his colleagues at Cambridge determined that amino acid sequence of insulin in the early 1950s. So that really was the start of determining how amino acids actually get folded and twisted into their unique shape. And this unique three-dimensional structure is what is going to occur under normal cellular conditions. And in almost every case, the function of a protein depends on its ability to recognize and bind to some other molecule, so this structure is really important. So, for example, an antibody binds to a particular foreign substance in our immune system, or natural signal molecules called endorphins will bind to specific receptor proteins on the surface of brain cells in humans, which produces a sense of well-being, euphoria, or relieves pain. So these signal molecules are specific proteins, and they have to be a certain shape so that they can bind to the receptor proteins. Morphine, heroin, and other opiate drugs can mimic endorphins because they're similar in shape, and they can actually bind to those protein endorphin receptors. So the function of a protein is considered an emergent property because it results from the specific molecular order of the polypeptide chain. Okay, so looking at that structure, we have three levels. We went over this in class, so I'll quickly review it here. We have the primary, secondary, and tertiary structures, which organize the folding within a single polypeptide. And this precise primary structure of the polypeptide chain is determined by inherited genetic information. So, of course, by your DNA, not by the random linking up of amino acids. Just a slight change in the primary structure can affect the protein's conformation and ability to function. Even just one amino acid change can make a huge difference. An example of this is with sickle cell anemia or sickle cell disease. There's a substitution of one amino acid which occurs from actually one point mutation in the DNA. So this substitution of the amino acid valine for the normal one, which is glutamic acid, at a particular position in the primary structure of hemoglobin, causes sickle cell disease, which is, of course, going to be inherited because it's that DNA mutation that has occurred. Glutamic acid is acidic and polar. Valine is not polar. So by being nonpolar, valine tries to hide from the water in the cell. And so what it does is valine causes the cell, the sickle cell, the blood cell, to actually turn in, as you can see in this picture. It misshapes the normal shape here because of that valine trying to get away from the water and that's surrounding it in the cell. So you can see here valine and glutamic acid, one being hydrophilic, one hydrophobic. So the abnormal hemoglobin actually crystallizes and deforms the red blood cells into that sickle shape, and they end up clogging capillaries and causing the blood not to be able to carry oxygen and to actually clog up the capillaries. Okay, so the next level of structure is secondary. This is the local folding. So we end up either a helix or a beta sheet occurring within a single region of that polypeptide chain. So that primary structure starts to curl, like in this helix, or actually fold. And you can have both of these occurring in one polypeptide chain, but they are going to occur in specific regions. So you'll only have beta pleats in one region, and then another region may have the alpha helix. These result from hydrogen bonds between the repeating constituents of the polypeptide backbone. So if you remember, the polypeptide backbone is the peptide bonds between the nitrogen group, the amino, and the carboxyl. And so it's actually the hydrogen atom that's attached to the nitrogen atom and the polypeptide bond that forms the hydrogen bonds. It ends up having an affinity for the oxygen atom of a nearby peptide bond, and that's where we get our, again, hydrogen bonds. And as you know, hydrogen bonds are weak, but the sum up of all these bonds stabilizes the structure of the secondary structure of a protein. This is another picture showing the two different types of folding or twisting. And some examples of this, like a fibrous protein like alpha keratin, is a structural